Hi, everyone. Hope you guys are having a great start of the Helix Summer Program. My name is Karan Linganeni, and to start this week off, we'll be going into a module for biostatistics. And specifically, we'll be defining what biostatistics is and going into some common biases and misrepresentations that you may see in conducting your own research study or analyzing one. So let's just get right into it. The first thing that we're going to do is actually analyze a case study, and that's John Snow's pump, which we'll actually be referring back to many times throughout these slides. So in 1850, the poor parts of London were actually going through a rough bout with disease, and specifically that disease was cholera at the time. It was not commonly well known that germs cause disease, but that's where Dr. John Snow came in when he noticed that the onset of cholera was almost always involved with vomiting and diarrhea as the most common symptoms. He therefore believed that the infection was most likely carried by something that people were eating or drinking, and therefore his prime suspect was actually the water that people were drinking, and he believed that it was actually contaminated by sewage. One of the things that helped him come to this conclusion was actually the map you see here to the right, and that is something that we'll talk about in the next slide. So Dr. John Snow was actually able to create this map using the deaths that they were recording during this time in London. And he was able to use this map to investigate for apparent anomalies. And these anomalies actually allowed him to implicate the specific water pump called the Broad Street pump as being a reason for some of these deaths. So the reason he was able to come to this conclusion was that he actually used the knowledge of another street pump, the Rupert Street pump, and the fact that many of the deaths were occurring in houses that were actually closer to this Rupert Street pump. It was commonly well known at the time in this area that even though the Rupert Street pump was closer to these houses, it was less convenient to get to because of dead ends and the basic layout of the streets. Therefore, the residents of those houses were actually forced to use the Broad Street pump instead. To further add to his conclusion, he found that there were actually no deaths just 2 blocks east of this pump. And that was the location of a brewery called a Line Brewery. And this is the place where actually workers drank what they brewed. And if they wanted water, they actually had their own well in which they took water from, which was most likely not contaminated in the same way and therefore did not cause cholera. So two of the questions that we were actually able to answer in these past couple slides is how Dr. John Snow was able to determine the cause of cholera and how he proved this hypothesis. I urge you guys to look back at the past couple slides and make sure that the answers to these two questions make sense to you. So going into what exactly biostatistics is, basically biostatistics is analyzing biological or health data using statistical methods. And essentially this helps solve problems in medicine, biology, or public health using these statistical methods. More or less, they help us find patterns in the data that we have. They help us determine causal relationships between variables that we may be researching, help us make inferences about total populations from our observations about sample populations, and they help us evaluate the best course of treatment or the best course of action when it comes to many of these problems within medicine, biology, public health, and et cetera. By using biostatistics, we can help determine these causal relationships, as we mentioned previously, and help aid public health issues like that of the cholera outbreak in 1850. So how exactly do we collect data and where exactly do we collect data? So going into the answer to this specific question, we need to define a couple of different terms. So specifically the difference between a population and a sample. So when we talk about a population or population size, we're talking about all members of a specified group. So for example, that could be all members of our nation, all people in the world. Specifically for this case study with John Snow, we're looking at all people who lived in London at the time in 1850. So all households in London. And a sample size in that sense was actually a randomized selection of maybe 25 houses or even more in different parts of London. So a very select few of those people or things that represented the total population, which in this case was London. Next, moving on to the exact way in which we conduct studies, we're looking mainly at experimental or observational studies. So when we think about experimental studies, we're mainly focusing on causal relationships and determining cause and effect between different variables that we're working with. And mostly in the realm of medicine or public health, that's going to be the course of applying treatments to a group and where we actually have control over these treatments. So in this case, going back to the case study, it's if Dr. John Snow was actually able to treat his patients with given cholera treatments possibly, and then basically determine the results of those treatments. Um, Next, moving on to what an observational study is. This is a study where researchers make basic inferences on what they've seen from data and the data they've collected. So this is, in a sense, what Dr. John Snow was actually really doing. He was simply using his data and then making observations and inferences about the total population from the data he collected using the maps. However, he wasn't actually applying any treatments or trying to determine any cause and effect in terms of applying a treatment and expecting a result. And the third thing is really not up for debate when we run these kinds of studies. Randomization is incredibly important when we run trials, no matter if it's experimental or observational. And this is something that really is a must. So a question that I pose to you guys is, are there any potential issues that you can think of that could affect the accuracy of this data that we see? Go ahead and think about that for a little bit, and we'll take a short break and come back to our next section. Hi, everyone. Welcome back from that short break. We're actually going to fall on and answer the question that we proposed in the previous slide, and that's going to be statistical biases. Specifically, statistical biases can affect the accuracy of the data that we get through a research study, and we'll talk about some of the very common statistical biases that we end up unfortunately seeing in many research studies. The first thing that we'll go into is a confounding bias, and that's when an unintended or additional variable actually affects the cause and effect relationship of the study that we're examining. So for example, let's say we're looking at the relationship between coffee and lung cancer. If someone was conducting a study on coffee and lung cancer, something that they actually might see is a positive correlation between that of coffee and lung cancer. Now, that's not to say that drinking higher amounts of coffee, in a sense, cause lung cancer. However, there's a hidden variable in all that, and that's smoking. It's actually well known that people who drink higher amounts of coffee are actually more prone to smoke. And of course, we all know that smoking and lung cancer are in a correlated relationship. Therefore, smoking, in a sense, was a hidden variable in this relationship and causes some unforeseen correlations between coffee and lung cancer. The second one that we're going to go into is a selection bias, and that's when results in a sample population are really not representative of the entire population that you're trying to make inferences on. Therefore, making any conclusions and trying to draw inferences on this total population are actually invalid. An example of this is, let's say Dr. Snow chose to use houses that were only on the south side of London, but chose to use London as the respective population from which he was actually going to use his inferences on. This would actually be a form of selection bias because he didn't take into account the other areas of London and the other households of London, and therefore making any conclusions from this study invalid. Next, we'll be going into a bias called a self-selection bias. And this is when a sample does not represent the entire population because it is self-selected, either in the form of polls, surveys. When done correctly, this can definitely be a good way of gathering data. However, when done incorrectly, it can cause a self-selection bias. So an example of this is, let's say Dr. Snow chose to poll to get his data. And specifically, he included a research description of what he was trying to get out of his research study, and specifically the effects of cholera, and trying to study how cholera was attributed to this pandemic in London. Specifically, something that we may end up seeing is that people who are affected by cholera are more likely to choose to take this survey, while people who aren't affected by cholera are more likely to push the survey to the side. So in a sense, this really is self-selected and would cause a self-selection bias. The fourth and last bias that we're actually going to talk about is the Simpson's paradox. And this is when an association between two variables in a population emerges, disappears, or completely reverses when a total population is divided into its subpopulations and that same study is run. So concept check for you guys. What type of bias do you guys think this research study is prone to? And take a quick second and we'll talk about the answer here in a second.